Okay, you guys ready? Stop. No? Yeah? Anyone? Over here? Recently, I've realized that the kids have taken over in my house. And, like, my husband and I, we were so tired because we both work. There's too many kids. And they'll just be, like, crazy, doing crazy kid things. And we'll be like, it's bedtime. I'm ready for bed. And they're like, ah. And sometimes I feel that way here because I'm kind of like, ready, teach you something. You guys are like, okay.
It's like having 90 kids.
It's like having 90 kids. Well, really, it's only like 20 kids or something. Okay. So as you guys saw, I changed the exam again. Sorry. I just don't know what to say about this transition from 3 days a week to 2 days a week is harder than I thought it would be. But that's okay. You guys have more time to study, more things. And then someone reminded me that we have like no lectures left afterwards for cell cycle, which is like tragic because I didn't use cell biology with like a crash course in cell cycle. But we're gonna do it. We're gonna make it happen, and it's gonna be beautiful.
No parachute needed.
Yeah. Okay, so stuff to teacher. The other day someone asked about regulation. What's the regulation of motor proteins binding to cargo? And we made that a stump to teacher, and I got a lot of very nice answers. I also got some people who answered a completely different question, so it made me wonder if it was clear what we were talking about. That's another story. I've only given a handful of points out so far, so if you haven't seen points come up on your blackboard, doesn't mean you didn't get them, it just means I haven't finished adding them. So basically what I learned, and I posted a really nice but old review on Blackboard, which explains a lot of this stuff. Well, they talked about how regulation, there could be two forms of regulation. One is in turning on or off the motor, which isn't really what we were talking about, that we were talking about regulation of actually binding to cargo. But when you talk about regulation in terms of binding of the motor protein to cargo, you're kind of making the assumption that as soon as you bind, you're going to move. So just to kind of realize that you can also regulate in terms of is the motor protein active or not. But one mechanism that came through, it's kind of what we talked about when we were speculating, is phosphorylation as a negative regulator, so inhibit binding or movement. And then there's the mechanism in kinesin, where there's this intramolecular folding, which we also kind of see, we're going to see with myosin too, where the motor protein is folded in such a way that its tail is inhibiting its motor domain and cargo binding. So in a way, you're inhibiting your binding because your tail is folded up and not available. And then also you're inhibiting movement because your motor domain is disrupted. And then when the cargo binds, there's a conformational change, which allows for the motor protein to work. So that's that. How do we feel about that? Who read the review I posted? Who reads everything I post? I just posted a nice thing that a student shared about some of this stuff with muscle contraction. So you guys look for that. Okay, here's our so far list. It keeps getting longer. So we've been through our structures, our actin, our microtubules. We've been through our accessory proteins, all these proteins that regulate the dynamic instability, basically, of these structures. We've been through, there's so much chit-chatting going on in this room today. It's really distracting. Is anyone hearing it, or am I losing my mind? Sometimes I think I'm losing my mind. Like, one of my kids was telling me her ears were speaking to her the other day, and I was like, it's not normal. That's not normal. I was like, are they telling you to do things? Like, I actually said this to her. I was like, are your ears telling you to do things? I didn't say telling you to do bad things. I just said, like, you're hearing voices. You've identified them as your ears. Anyways. Okay, so we went through all these. We went through the motor proteins, and that's kind of where we've stopped. And so now we're going to start more of the putting it all together stuff. So this is where we stopped on Wednesday, right? So you can think of an interface cell, at least, as kind of like having this microtubule highway upon which things can travel. And your minus ends are at the center of the cell in your centrosome. Your plus ends are at your plasma membrane. Wow. So your dynein is going to be moving towards the minus end. So anything that needs to be coming inwards will be traveling by dynein. And your kinesins mostly move to the plus end. I'm saying mostly because not all of them are plus end traveling, but most of them are. So it's okay to kind of just generalize there. So what motor protein would be involved in transport of a vesicle from the Golgi to the plasma membrane? What motor protein would we use? Okay. So this kind of is involved with our stump the teacher question a little bit. So how do motor proteins transport cargo? So I tell you they do. You know they walk along the cytoskeletal structures, but they have to bind to their cargo in some way. And so this is from that review that I posted. And I'm sure this is 2003. I'm sure we can find a nicer, newer one. But I like this figure. And so this is showing the cargo, the motor protein, and the structure, microtubules, or actin. And you can see they mostly have to do with some kind of transmembrane protein that's in the organelle or in the vesicle. So there'd be a transmembrane protein either directly associated to the tail of the motor protein, or associated through a linker protein, or associated through a complex of proteins. And then there's actually also a way to associate directly with phospholipids in the membrane. And so this is true for both kinesin and dynein, except they're just a little bit different. Dynein seems to use more complexes of proteins, and we'll have an example of that. And then myosin also similar, where you're associating with some protein or protein complex that's associated with the vesicle or organelle being moved, or associating with the phospholipids. Okay, so that didn't really talk about regulation, but then if you throw in that level of phosphorylation or conformational change, it starts to kind of come together. Any questions about this? Yeah.
What was the list of different proteins? Did you say linker protein? Like when you're laser pointing across the top?
Oh, so you can kind of just look at it and think about it, right? So this thing that's going through the membrane is a transmembrane protein. And then there are these linker proteins. So it's a protein that's kind of linking your transmembrane protein to your motor protein. Or it could be a complex that's acting to link your transmembrane protein to your motor protein. Either way, it's all about interacting with your motor protein and what exactly is interacting. Yeah. Any other questions? Okay. So for kinesin, the motor protein tail or light chain, it attaches to cargo through these membrane-associated motor receptors. So kind of, these are these membrane-associated motor receptors. Okay, so cargo, it needs to go to different places. So now you should start just thinking a little bit about what we did in the beginning of the semester, where vesicles needed to go to certain places, right? And so you could imagine this cargo also needs to go to certain places. So some of the cargo might be vesicles. So what determines where things are going? So you're going to have a membrane-associated motor receptor, and you're going to be interacting with your motor protein, but what's going to determine where it's going? So you're talking about what track is it walking on. So I just heard, oh, the motor domain. The motor domain determines the track it's walking on, but what about its final destination? What did I hear over here?
I just said it was a bug moving really close to you. I don't know what it is.
Ooh, what is it? Oh, it's one of those jumping crickets.
Oh,
Right? I learned spiders. Spiders have 8 legs. Who's an entomologist? It's not a spider. 1, 2, 3. Only 6 legs, not a spider. Who's here? Guys, listen, okay, ladies in the room, please do our future generations a favor by not being creeped out by bugs. Because, I mean, men are like, we're always like, get the man, kill the bug. Like, we're so big. We're small.
That's hard for like he chased me.
He chased
me.
I don't care.
He did. That's like
I don't care.
Okay, let's get things back under control. I'm not going to kill the bug because it's not going to hurt me. Who feels me need to kill the bug? Raise your hand.
Get outside.
Okay, listen, guys. It's going to be okay. It's going to be okay. I will protect you with my motor proteins. My stories about motor proteins. It's going to be okay. If it attacks me, everyone have 911 on your phone. For the last one, if it attacks me, it's going to be okay. But thank you, guys, for having my back. Huh?
I said, do you want me to take it outside?
You can, but it's okay. I think it's okay. Maybe he wants to learn.
Oh.
Here, why don't you go over there? I think have I already stepped on it? It's injured. It's not going anywhere. I just tried to make it jump. Anyways, jump. How is Carvel going to know where to go? Okay, that's the question.
Would we tag or something?
Will be tagged. Well, it's okay. So you have a vesicle. And it contains things that were already, so we were ER, remember ER, Golgi, wherever. You're in a vesicle. And now that vesicle needs to go along some motor protein to get to where kinesin is going to pick it up and take it to the plasma membrane. Or take it to wherever. But how, can we remember how it knows where to go? Yeah. So I'm hearing, so you guys are like, you're on it. Yeah, there are signals that take things to certain places, but remember, those are cytoplasmic proteins that gain entry to the nucleus. So, yep.
Is it like RAB interaction?
So RABs, right? So I'm trying to get you guys. Did anyone come across RABs when we were talking about regulation? When we were doing stuff with teacher research? I came across RABs a little bit. No, yeah. Okay, so remember we had RABs, and what else did we have? RABs and snares, right? So let's like try to put it all together. So now you're going to have a vesicle, and it's going to have a membrane-associated motor receptor that's going to bind to motor protein, and it's going to walk along. But it's also going to have, woo. See, I thought I saw jumping at me. That girl went crazy. You're in the corner. Oh, over there. Okay. You're getting nervous. Okay, so good. Grabs. Let's bring it back. Bring it back. Grabs, snares, Gaps, no.
Oh.
Okay, you guys are getting crazy. Yes, gaps. You're right, gaps, deregulate rest. Okay, so where would we find these motor receptors?
Everywhere.
Like they would actually, they're membrane associated, so remember we're moving our organelles and our vesicles, right? So they're actually integral membrane proteins on these vesicles or organelles. So so there's these proteins that are called J and K interacting proteins, or GIPs. And they're scaffold proteins that are involved in cell signaling that function as kinesin receptors. Whoa. Who's going, whoa? Whoa. Right? So that's kind of cool. So there are these, remember J and K, these Janus kinases that are in signaling? Who already forgot about signaling? Nobody. You guys all remember it. So these proteins are also kinesin receptors. So you can kind of imagine how this might link transport and signaling, right? With signaling, you have all this stuff moving around in the cell too, right? You're having kinases and you're having endocytosis. You have a lot of things are induced through signaling, right? So you can kind of imagine how they might need to be linked. So with dynin's attaching to membrane-bound cargo through the dynactin complex. And so this is a complex of proteins that contain, so you've also got some kind of membrane protein that's going to interact with some other protein that's going to interact with an actin-like protein. So it's a whole complex of proteins that make up this dynactin complex that that connects your vesicle to dynin, and so can walk along the microtubule. This is not actual actin, it's an actin-like protein that's made of ERK1, which remember has a similar structure to actin. So this is how vesicles can walk along microtubules with dynin. Okay. Any questions? No more bugs? I think we're okay. No snakes? Okay. Okay, so we're talking about how motor proteins interact with their cargo. So myosin 5 transports melanosomes to the ends of the dendritic processes in melanocytes on actin filaments. So what are melanocytes? Does anyone know? Who wants to guess? Something to do with melanin? Yeah. They're the cells that make melanin, and they're stored in melanosomes. Remember we talked about lysosomes that have specific functions, and one was melanosomes. So these specifically store melanin. And so the melanocytes make the melanin, and they're stored in the melanosomes, and they need to travel to the ends of these dendritic processes in the cells, and then they're delivered to neighboring keratinocytes, which make up your skin and give you skin color. And so these melanosomes these are membrane-bound organelles that contain melanin, and they're transported by myosin 5 along actin. And so this is actually showing, this is microscopy where each of the dark spots are showing the melanosomes. And then I believe this picture was immunofluorescence staining myosin 5. And you can see how they completely overlap, right? Suggesting they're in the same place. Right? So myosin 5 is transporting melanosomes. So what do you think an individual with a mutation in the myosin 5 gene might look like? Yeah?
Isn't that for albinism?
is that where albinism comes from? I'm not 100% sure if it's albinism, but it might be. But definitely they will have, so mice that have a mutation in, I guess it depends on the mutation, in myosin 5 have a diluted color to them. Yeah, so you'll be lacking skin color. So if you're thinking about myosin 5 and a mutation that might result in albinism or decreased skin color, where do you think, where on myosin 5 would you expect this mutation to be? Yep.
Tail domain that binds to the melanosomes.
Tail domain that binds to melanosomes, right? Well, so I guess you can think both of them, right, in a way, because as I was thinking about, as I was asking the question and coming up with the answer, I was thinking about this. Yes, the tail domain binds to the cargo, so if it can't bind to the cargo, then you can't move your melanosomes, so they can't get delivered to the keratinocytes. Right. But what if there was a defect? What if there is a defect in the motor domain? Would that have the same effect, or do you think that could have a different effect? Or what about an effect on the individual who has this mutation?
Same.
So it would have the same effect on their skin color, but do you think the individual might have other issues too? Probably because myosin 5 does a lot of things, so if myosin 5 suddenly can't work at all in the cell and it can't walk along actin at all, then it might have many issues. The individual might have many issues, whereas if it's some kind of specific interaction between the tail and the cargo, maybe it would be more of just a skin color issue. I don't know. Things to think about. Lovely discussion for Monday afternoon. Yeah. Okay, good. Okay, so myosin 5. Another thing myosin 5 does, which is why this person might have a few issues if they were yeast. So it's involved in mRNA transport. Okay, do you ever think about mRNA and how it gets to where it needs to go and these kind of things? Right? So mRNA needs to associate with the ribosome and be translated, but also sometimes mRNA kind of gets localized to a certain part of the cell and then also associates with the ribosome. Translated, but it's in the right location for where that protein will be needed. And so this is kind of talking about that. So this is in yeast. And so there's this ASH1 mRNA, and it only needs to be expressed in the daughter cell. And so what happens is when the ASH1 mRNA is made, it binds to, it has a region that binds to this RNA binding protein called SHE2, which then will associate with this linker protein and interact with myosin 5, and it travels over to the growing bud, the location where the daughter cell will be budding from. This is budding yeast. So that way, when the daughter cell buds, it'll have that RNA present in that cell. So here you're having the cargo is RNA. This is not a membrane-bound anything. It's just RNA, and it's interacting with the motor protein through 2 different proteins. Any questions about this? So this is just showing, this is FISH, which is fluorescent in situ hybridization. So you can make a fluorescent probe to whatever RNA or DNA you're looking at. And here's a probe made to ASH1 that happens to be red. And so you can see ASH1 is right here. And so these are 2 different pictures of the same thing. One is looking at fluorescence and one is looking at the cell. And so here's the bud tip from the yeast. And that's exactly what happened, is ASH1 mRNA localized right up here in the bud of the yeast. So if you wanted to inhibit this from happening, what would you mutate? Yep.
Region 5 prime region.
So right, so you can exactly you can mutate this region here on the RNA. What else can you mutate? You can mutate your sheep protein. What else can you mutate? What about on your myosin? Your tail, right? Exactly. So if you made any of those mutations, basically that inhibit the interaction of your RNA with your myosin, then you're not going to get localization. Okay, so you can regulate myosin activity through phosphorylation. So there's a kinase called MLCK, and it regulates bipolar filament assembly. So that's assembly of myosin into a bipolar filament that looks like this. So that's where you get this coiling around the tails to organize them so that you participate in contraction. And so when you're inactive, when the myosin is inactive, it's folded. And when you have phosphorylation, then you get a conformational change which releases the tail. And that allows, so your light chains up here are phosphorylated, and that allows assembly in your bipolar filament. And this is involved in the kind of contraction so this is myosin 2, and this is involved in the kind of contraction that you're going to see in your contractile ring during mitosis. So it kind of makes sense that MLCK is activated during mitosis, because this allows for assembly of myosin 2 into your contractile ring. Isn't that cool? So I guess something to think about, and something that we can think about as we go into cell cycle, is what turns on MLCK. So some of you guys have started thinking about cell cycle because of recitation. And so if you need to activate a kinase, you can activate a kinase probably by a few different things, but one of the more common things to do is either phosphorylate the kinase or dephosphorylate the kinase somewhere, so you can regulate it that way. And so you can imagine that some kind of kinase is going to activate this kinase. And since cell cycle is all about regulation by CDKs, cyclin-dependent kinases, you can imagine how maybe, and I don't know, I'm just reasoning, maybe MLCK is activated by a mitotic CDK. So let's think about that and come back to it when we talk about cell cycle. Okay, so here's another example of regulation. So fish melanocytes can alter the location of their pigment granules in response to a neuronal hormonal stimulation. And so you can either have dispersed melanocytes, which gives you this kind of appearance, or you can have aggregated melanocytes, so they're all in the center at the minus end. And this is regulated through phosphorylation of the kinesin light chain in response to a signal. And it then activates the kinesin, which allows dynin to take over. So dynin moves to the minus end, so you have kinesin, which is going to the plus end, and then you get phosphorylation, so that allows dynin to take over. So what do you think the phosphorylation is doing? Yeah.
Conformational change.
But really, what is it doing? So I heard conformational change. So conformational change of what affecting what? Yep. Is it preventing the kinesin light chain from interacting with the melanosomes? Maybe. So it's phosphorylation of the kinesin light chain. So what part interacts with the melanism? Not the tail. But either way, so interaction with cargo, that's where you're going with that. Right, so if you're preventing interaction with the cargo, which in this case is the melanism, that frees up the cargo to interact with dynin, which will move the melanisms back inwards to the minus sign. Okay. Do I have to teach muscle contraction? I always feel like it's so redundant because so many of you guys have learned it. Can we skip it? I'll do it. Okay. Skeletal muscles formed by the fusion of multiple myoblasts. They contain many myofibrils. So those are these things. And this is where your actin and myosin are participating in your contraction. So remember, they're made of multiple cells that fuse together. So you can see that there's multiple nuclei. So you've got red stained myofibrils, that's where your actin is. You've got the blue stained nuclei. If I asked you how many cells made up this muscle cell, you would tell me a lot. Don't count them. So your myofibrils are made up of these repeating contractile units called sarcomeres. And your sarcomeres are made up of thin and thick filaments. Your thin filament is your actin, and your thick filament is your myosin. And so here's one sarcomere right here. And so your dark band is your myosin, so that's your thick filament, and your light band here is your actin, which is your thin filament. Your Z disc is where your plus ends of your actin are attached. And then your M line, which is right down the center here, is where your adjacent myosin 2 filaments are joined. So this is a better picture. And so here you're seeing your myosin, which is assembled into your bipolar. So it has 2 ends that walk in opposite directions, right? Thick filaments. Then you've got your actin, which is capped at one end by cap Z along your Z disc. Remember we talked about cap Z. And at the other end by your tropomodulin at your manus end. And then there are these molecular rulers. There's nebulin, which determines your actin filament length. I think it's supposed to be one of your largest proteins. And then titin, which is a molecular spring that positions your thick filament, so your myosin, midway between your Z discs. So the length and spacing of your sarcomere are dependent upon a number of these accessory proteins that we've learned about. Questions? I'm guessing everyone knows this already. No. We'll see. Okay, so here are the basic steps in skeletal muscle contraction. You get some signal from some motor neuron, and then there's an action potential triggered in the plasma membrane, which spreads to your T-tubules, which then there's a signal that's relayed to your sarcoplasmic reticulum, and then the magic happens, which is that the calcium channel opens, and calcium comes out of your sarcoplasmic reticulum. Calcium floods the cytosol and initiates contraction of each myofibril simultaneously. And then to turn it off, the calcium's pumped back into your sarcoplasmic reticulum, and you get relaxation. There's my physiology. Right, so your sarcoplasmic reticulum is this kind of netting kind of looking thing that's surrounding your myofibrils, and that's where your calcium is stored, and when you get a release from there, that initiates contraction. So what I care about is, what is the calcium doing? What is it actually doing on the molecular level to initiate muscle contraction? You guys know this, so tell me. I can teach it to you, but I figured I'd just have you guys teach it today. I feel like teaching. No, it'll come in another slide. What's the calcium doing? Someone raise your hand. Don't make me comment. Yeah.
Isn't it binding tryptomycin to uncover the actin heads?
So is it binding to tryptomycin to uncover the actin, where the myosin binding site's inactive? Close. It's affecting tryptomycin positioning. What did I hear? Troponin.
Causes a conformation change in tryptomycin, and the actin site, or the ATP sites on the actin don't open, so the little heads can get.
Right. So there's this. So you're close. So your tropomyosin is covering where your actin's going to bind, and it's being held in place with this troponin complex. When calcium comes and binds, it releases that complex, allowing your tropomyosin to move out of the way, so you can get interaction between your actin and your myosin. So this is what we're talking about, right? So you have your actin, and then you've got your tryptomycin. Remember, that binds along the sides and stabilizes your actin filament. And it's being held in a certain position, which is blocking interaction with your myosin by this troponin complex, which is made of 3 different parts. The I, which is the inhibitory part, the C, which is the calcium binding part, so this is where the calcium comes into play, and then your T, which is your tropomyosin binding, so this is what's holding your tropomyosin in place. And so when you're resting, your troponin IT complex is in the way, it's holding your tropomyosin in the way of your myosin 2. And so when you get this influx of calcium, then it will be bound by the troponin C, and that releases the I, which allows the tropomyosin to slide out of the way. And so then your actin's available for myosin binding. And so this is kind of showing how it would look in a cross section. You have your actin, and then your tropomyosin is along the side, and it's blocking your myosin binding site. But when you add the calcium, the troponin IT complex releases and allows the sliding of your tropomyosin out of the way. And so you have the ability for the myosin to bind. Okay? Any questions about that? So calcium binding by troponin C results in release of troponin I, allowing movement of tropomyosin out of the way, and muscle contraction can occur. So this is showing it again, how you have your tropomyosin kind of covering. It looks like olives here. I like that one. Who likes olives? I promise I will not talk about food during this class. I'm sorry. But we got one back there liking olives. That's good. So the tropomyosin is covering the olive holes where you put your fingers. It's like a pimento. And so then when the calcium binds, then the troponin complex moves, and that slides the tropomyosin out of the way. Okay, was that fun? Yeah, you guys got a little brain break there. Okay, so let's talk for a minute about cilia and flagella. So these are motility structures that are built from microtubules and xanthine. So we talk about all these different cytoskeletal structures, and we talk about how they can be dynamic, and then we also talk about some structures that can be very static, right? like, so muscle was an example of a very static structure. Yeah, you're gonna have like the change in terms of interaction, interaction between your actin and your myosin in the presence or absence of calcium and all that. But really, your muscle cells are not like growing and shrinking. Your sarcomeres are staying pretty much the same. Well, so also your cilia and your flagella, those aren't changing. Their movement might change, but their length, those microtubules, are not changing in length, just like the actin in your muscle cells is not changing in length. So this is another kind of static structure. So you've got your flagella, which is used for swimming on sperm and protozoa. And then you've got your cilia. And this is, you can see this on your respiratory tract or moving eggs through the oviduct. Also, so it's about moving fluid, so you'll have a lot of cilia moving fluid across the tissue. And so these are built for microtubules and dynin. So the core, or the axoneme of cilia and flagella, is composed of these microtubule doublets that are arranged in a ring around a central microtubule doublet. And you have dynin, ciliary dynin, that binds to neighboring microtubules. So there's this interaction, between you've got your microtubule doublets, which are kind of connected, and then you've got this interaction between your dynin that's attached to one microtubule and moves along another. So ATP hydrolysis is resulting in walking to the myosin, because this is dynin. So you're walking to the myosin of one microtubule while you remain bound to another. So that's what causes the bending motion that you see in cilia and in flagella. And so this is how it looks. So you've got these two. So this is, you can imagine here's two doublets that are next to each other. Like these two right here. One doublet, two doublet. Oops, that's not what I wanted. And so you can imagine if they weren't attached in any way, and you're bound at one end and you're walking to the myosin, you're going to slide upwards. But in flagella, they're actually attached by these linking proteins, so that when they walk to the myosin, you get this bending. And so it's that bending that causes the movement that allows for sperm to swim, or cilia to do their thing. So if you're thinking about ciliary dynin and their function in movement in the flagella or in cilia, what would be the outcome if there was a defect in ciliary dynin? Yes?
Your cilia wouldn't be able to move like fluids like mucus and stuff like that.
So you could have issues with mucus, right? So in your lungs, your cilia, and where else? Other places? You wouldn't be able to move mucus? Okay, so that's one thing. What else? What else wouldn't you be able to do? Yeah. Right, sperm wouldn't be able to swim. Exactly. So there's this Hurta-Jenner's syndrome. I'm not sure if that's the right pronunciation. And those are two of the things that these people have. One is male sterility, and then also lots of upper respiratory issues due to the inability to move mucus in their lungs, the respiratory tract. Okay, so we've been talking about these cytoskeletal structures. So you can think of two things. One group where there are these structures that are constructed when they're needed. And then we can talk about structures that are always there, right? So these myofibrils, cilia, flagella, they're always there. And then others are constructed only when they're needed. in response to some kind of signal. So what are some examples of structures that are only constructed when they're needed? One specific structure, yes.
Contractile ring.
Contractile ring, so that's one structure that's only constructed at what point of the cell cycle? Metaphases, exactly. Trying to give away some easy ones since we have quiet room today. What other structures are constructed only when needed? Think microtubules, yes. Mitotic spindle, which is only constructed one. Interphase, anaphase, not an anaphase, but maybe starting at prophase. Starting at prophase, metaphase. Yep. Good. Right, so your microtubules and motor proteins are required for construction of mitotic spindle. Are you guys cheering back there because you were guessing? And you were right, and you're so excited? That's awesome. I love to see people cheering. The back rows, specifically. I always worry the back row is full of people Facebooking. You guys aren't Facebooking back there, right? Everyone's like, no. Okay, how about looking up recipes for dinner? Laundry list? Nice. Okay, good. You guys are all working hard. Trouble. Okay, mitotic spindle. Okay, so the interphase microtubule. So here's interphase, right? And here's your mitotic spindle. So these microtubules need to be rapidly disassembled and then reassembled into bipolar mitotic spindles, right? And this occurs in under an hour in most animal cells. That's pretty fast. I thought it was even faster than that, honestly. What proteins do you think might be involved in this, going from this to this? We're just guessing here. What kind of accessory proteins do you think might be involved in the rapid disassembly? Actually, let's give you guys a break. I want you guys to think about it for a minute and come up with a mini list of proteins that you think might be involved. And it doesn't have to be right, it just needs to be logical. That might be involved in the disassembly of your interphase microtubules and then involved in your assembly of your mitotic spindle. Okay, so take like 1 minute, talk to your friend, discuss. Okay, guys, what do we think? What do we think? That was your minute. What do you guys think? Yes.
The severing protein on to break up all that.
So you use the severing protein to time. That's one. I don't know, and I haven't done any experiments or research, but yes, that's what I would pick. What else? We just came up with Katana, that's it. Anything else?
Don't certain maps destabilize multi-pages?
Down some maps. Yeah, I came up with a map. Which map did we come up with? Kinesin 13 catastrophe factor. I thought that might be one. Yep, and I came up with one other one that might encourage disassembly. What other one do you think might encourage disassembly? Yeah. Staffman. Yeah. Yeah. And so then I was trying to think in my head what might encourage. So then you're going to have in prophase you'll get your duplication of your centrosomes, and then you'll have them growing again. And so I was trying to think, well, what will encourage that? What did you guys think? Huh? Yeah. Kinesin is a motor protein. So motor protein will be involved in movement of your mitotic spindle to where it needs to be. What do you think might encourage growth? What's going to nucleate? Yep. Gamma tubulin ring complex, which will be a part of your centrosome. Were you going to say that? Yeah. So gamma tubulin ring complex. And then I was kind of thinking to myself, well, we just disassembled, and based on just the kind of rules of encouraging growth, when you have a lot of subunits, we should have a lot of subunits. And if we've nucleated, then they should just grow, right? Everyone, you guys just think of that. Because I don't know if you really want some stabilizing proteins there, because you're kind of very dynamic, right? Or no? Yes.
Would the Italian work for this one too, since it's very
Right, but think of it as going, so the comment was, wouldn't Katana work for growth also since it's creating more subunits? And that's kind of what I was trying to say, was your Katana-induced disassembly creates the subunits that allows for the reassembly. Yeah. Okay, any other thoughts about this? Everyone's happy? Okay. Okay. So this goes to one of the quiz questions for the weekend. Interphase microtubules undergo rare catastrophe. So they're pretty stable. They're not undergoing catastrophe very often. What kinds of regulatory proteins are likely to be bound to interphase microtubules? So you guys can tell me what you think. Yes?
Oh no, wasn't it? Oh, do you not want me to
Go ahead.
Oh no, wasn't it XMAC 215?
That was what is, yeah. Sorry, I was confusing myself there for a minute in my head. So the answer was XMAP 215, and I apologize for those who got confused by my wording early on. Don't worry about that. A few people emailed me, and there was a typo in there that was throwing people off. So the first few people who took the place, the go-getters who took it first, kind of got confused. Right, so XMAP 215 is stabilizing or destabilizing? Right, so it's kind of logical that if there's not much catastrophe going on, then you're going to have something stabilizing probably involved. And so what will you probably not see? What kinds of proteins would likely not be bound? Kinesin 13, what else? Anything else? Staffman? Probably not, right? Because that would induce catastrophe, right? So how would you answer this question in the lab? So let's say you were studying. So we just developed hypotheses, right? That's what we just did. We discussed. We thought. We used our prior knowledge to come up with a list of things that we would expect to be bound or not bound. So what would you do if you were testing this in the lab? Keep it simple, because you guys like to get really fancy. What would you do in the lab? Do you want to talk about it? Do you want to think about it for a minute? Why don't you think for a minute about how would you, so we came up with, what did we come up with? We came up with, we would see X215, but we would not see Hi, Nice 13, or Staffman. So think for a minute to discuss with a friend how do you test this in the lab? What is there and what's not there, and what the effects are? Or that the other Okay, guys, what did we come up with? So the question was, we've decided that XMAP 215 should be present on interface microtubules because they're not really going over catastrophe, they're going through catastrophe, they're fairly stable, and that kinesin 13 and Staffman would not be there. So what could you do in the lab that's fairly simple to determine whether or not this is the case? Test your hypothesis. what did you come up with?
Mutate the map.
Mutate the map.
The X map, where I can't read from here.
Yeah.
Glasses.
And what would that
mutate
do?
it and make it inactive.
And what, okay, so mutate XMAP 215, make it inactive, and see, and what do you expect would happen?
Well, if you're expecting that to be there to be the stabilizing factor if it's inactive, it would
So-
or some funky stuff about. That's a type of.
Mutate it and look for funky stuff. Okay, mutate it and expect more catastrophes to happen. Okay, so that would be like experiment 2. What would experiment 1 be? You don't even know if it's there. So do you want to go to the trouble of mutating it? Yes.
Do we know that Kneeson and Staffen are there?
We don't know anything. We're hypothesizing what's there. Yes.
Tag it,
So tag XMAP 215, tag your microtubules, or use an antibody that's fluorescent, either way, and see whether or not they're at the same place. So it would be something like doing immunofluorescence. Is that what you're saying? Yep. So you would stain one green and one red, and then see if you've got some stuff that's yellow. Did you have something? Anyone else? So you could do that, see if it's there, then you could do your experiment, mutate the binding site, see if that affects catastrophe. Good. Good. And what about for the kinesin 13 and the Staffman? What would you do? Yes?
You just assume that's antibody. You're not about to assume that's antibody. So they won't get functional.
So use some kind of inhibitor. So assume it's there and you put some kind of inhibitor and see if some function changes.
Yeah, like, so if you're considering condensate or Staffen and if it doesn't, like, say it's multi process, this can't do that.
Okay, so using it kinesin or Staffman inhibitor and see if it stays stable.
Yeah.
That's a hard one since it's stable. But I see what you're trying to do. Yeah.
After tagging them, are you able to isolate them and then watch them on their own, or do they all have to be in the cell they're acting somehow? Like
What are they?
the kinesin, the XMAP, and the Staffen.
You would have to do everything separate. It would all have to be separate. So you could do an immunofluorescence assay where you have an antibody against XMAP 215, and you would look to see if it's there. You would look at your microtubules. You can get another antibody, specifically look for Staffman, see if it's there, if it's binding to lots of subunits. Look at kinesin 13. Now you can say kinesin 13 and Staffman are really most likely not to be there because of the stability, or not to be present in high amounts. Yeah. Okay. How are we doing? Are we satisfied with that? A little bit. Who wants to come to the lab and do it? Come. Everyone. Okay. Huh? Okay. So, motor proteins are very important in positioning of the mitotic spindle. So this is from a paper, a review, and it's just showing that there are all different kinds of motor proteins that are involved in organizing the mitotic spindle and getting it to where it needs to go. And so you've got these bipolar plus end-directed motors. So there are these kinesins and these dyneins, and there's all these different structures of them, and they all move in different directions. So for example, this one is going to be helping with positioning of the different poles. These out here, these motors are going to be partially binding to microtubules, partially binding to the cortex, and helping to position the mitotic spindle in relation to the actual periphery of the cell. So this is another picture. So there's actually 3 different types of microtubules that make up your mitotic spindle. There are these interpolar microtubules. These are the pink ones. And they're just interacting with each other. And the circles in between are the motor proteins. And they're involved in the positioning, the moving in and out of the spindle to get them to the actual poles. And then when you have anaphase, they're involved in pushing the poles apart. And then you have your astral microtubules, and those are pushing against the plasma membrane, and those are also interacting with motor proteins that are bound to the plasma membrane or the cell cortex. And then you've got your blue kinetochore microtubules that are interacting with the kinetochore and your chromosomes. And those are the ones that are going to be involved in chromosome segregation. Okay, so this is a picture that I totally love because it's kind of putting together a lot of what we've learned into one picture and one process. And this is, so I call this the big picture. This is actin and cell movement. So it starts, where's A? Well, it starts over here where you've got some kind of stimuli coming from outside the cell. And that's going to tell the cell it's time to move. And so you're going to get signaling that's going to activate WAS, right? And that's going to activate ARP. And then that's going to, so they're going to go and they're going to start nucleating actin. And so you get nucleation and you get these growing of these actin filaments that push the membrane upwards. And so this goes back to that initial paper that we did with the E. coli where it was forming this pedestal by inducing recruitment of ARP and WASP and nucleating actin and pushing the plasma membrane up to form the pedestal. So you've got all this ATP actin that's bound to profilin. And it actually, so you have two things happening. So you've got the actin is being added, and you've got ARP, and you've also got somewhere you should have hormones. Nope. And so then you're getting addition of your actin, and you're moving forward. But then in the rear, you have cofilin. So if you guys remember cofilin, it specifically targets ADP actin. And so you've got your cofilin coming at the rear and disassembling your older actin. And so that creates more subunits that can then bind to profilin, and then they're available to be added to the growing actin filaments. And so then you can have some capping proteins that might stop elongation, but if you're moving forward, you're going to have that constant elongation, and then you're going to have the disassembly of the older actin and reassembly of new actin and pushing the cell forward. So this is where we're going to stop today, and hopefully we'll finish on Wednesday. Oh wait, guys, 1 second. So I had originally said I needed multiple choice questions by Tuesday. You can get them to me by Thursday.